Hello my dear student, তোমাদের সবাইকে welcome. So today we discuss a very important question about the cell chapter, the fundamental unit of life. So today we discuss the most important some questions from the most important chapter, cell, right? At first we are discuss about the 1 marks question, after 2 marks, 3 marks and 5 marks question, right? So one by one we discuss all of the most important question about the cell chapter. So already we know that cell is the fundamental unit of the all living things, right? You know the prokaryotic and eukaryotic cell is there. In the prokaryotic cell their nucleus is not developed. On the other hand, the eukaryotic cells, you know, their nucleus have that membrane bounded structure is there. Also some membrane bounded cell organelles are present, and the example like the plant or animal cells are the example of the eukaryotic cell, right? So that first question, the plasma membrane. Plasma membrane is made up of which of the two components? You know that plasma membrane, also it is called the cell membrane. They are covered all of the cells, plant cell, animal cell. In case of bacterial cells also they have the plasma membrane. This plasma membrane, the main composition or chemical composition is the phospholipid, right? Phospholipid, right? And then the question is there, the two components. So lipids and the protein are the two components. This is present in the plasma membrane structure. Next we are discuss the second question. This is the hypotonic solution. You know the different concentration, according to the different concentration the solution is the three type: hypertonic, hypotonic and the isotonic type of solution. But how you define the hypotonic solution? That is the solution having the solute concentration lower that of the cell sap. That time it is called the hypotonic. So a solution having solute concentration lower that of the cell sap, like you place a cell in a beaker. You place a cell in the beaker, right? That time inside the cell sap have some concentration and outside the cell have some concentration. But in the hypotonic case, the solution having the solute concentration lower that of the cell sap. That time it is called the hypotonic solution. This is the most important question in the CBSE class 9. All the students please bear in mind. Next question, what is the hypertonic? Hyper means the more, right? That time the solution having solute concentration higher that of the cell sap. When the outside the concentration of the solute is much more higher than the cell sap, that time it is called the hyper. Hyper means the more concentration, more solute concentration, right? Okay. Next question, what is the isotonic solution? What is the isotonic? Iso means the similar, right? Right, in that particular type of solution having the solute concentration same as that of the cell sap. Same example, like you place a cell in the beaker and the outside the concentration and inside the cell have the same solute concentration. That time it is called the isotonic solution. These three important questions define hypertonic, hypotonic and isotonic solution. This is the suggestive type of question. That means this question is the most important. Please you bear in mind. Next question, question number 5. Next question, cell wall is made up of which component? I think you already know it. What is the main chemical composition of the cell wall? What is it? This is the cellulose. You know the cellulose, it is the monosaccharide, polysaccharide or disaccharide. If you know, please you write down in the comment box. Cellulose, this is the chemical composition of cell wall, but cellulose is the polysaccharide, monosaccharide, or the disaccharide. If you know, please write down in the chat box. Cellulose, this is the monosaccharide, polysaccharide, or the disaccharide. Which one is the right option? Please write down in the comment section. If you are no, please write down. Cellulose is the polysaccharide, right? Okay, now we go to the next question. Give an example of the unicellular organism. Unicellular means they have only one single cell, right? Single cell organism, also it is called the unicellular organism. Like amoeba, like bacteria, like paramecium. Anyone, you write down in your copy. All is the example of the unicellular organism. But you know, the amoeba, bacteria and paramecium, which one is the prokaryotic cell? Please write down in the comment box or the chat box which one is the prokaryotic cell according in that particular three example I given you, amoeba, bacteria and paramecium. But which one is the prokaryotic? All of these three example is the unicellular organism, but which one is the prokaryotic? Please you write down in the comment or chat option. Okay, next question. I tell you answer. The bacteria is the prokaryotic cell. The amoeba and paramecium are the example of the eukaryotic cell. But three example are the unicellular organism. Now we go to the next question. Give an example of the multicellular organism. Multicellular means they have multiple number of cells in their structure, right? So this is called the multicellular organism. Like the human beings, like the fungi, like all plants, like all animals are belongs to the multicellular organism, right? Now we go to the question number 8. Now we are go to the question number 8. What is the active transport? What is the active transport? That means how you define the active transport? The movement of molecules across a membrane in the cell against a concentration gradient with the help of ATP. It is called the active transport. Like one membrane is there. Some solute concentration in that particular A site is the low. On the other hand, the B site in that particular section or part, the concentration of the solute is much more. But in here, the solute are transported from the low concentration to high concentration with the help of ATP. That time it is called the active transport. So please you bear in mind, active transport means in here the ATP are involved and this is happening against a concentration gradient. Clear? Now next question. What is the intracellular source of the digestive enzyme? That means intracellular, that means inside the cell where the digestive enzyme is there, right? Digestive enzyme is present in our stomach, in the small intestine. Okay, but in that particular question, what is the intracellular? That means the digestive enzyme present inside the cell. Which cell organelles is there, right? This one is the lysosome. Lysosome is the most important cell organelles which is filtered up the digestive type of enzyme, right? Very important question. Next question, what is the endocytosis? Endo means the inner, right? Endocytosis is the ingestion or engulfment of the food or other material by the folding of the plasma membrane. You know, like amoeba. This is the structure of the amoeba, and in that particular amoeba, it is the food, and they engulf this food. They engulf this food by this help of the plasma membrane, folding of the plasma membrane. This is called the endocytosis. That means the food are enter inside the cell. Endo means the internal, inside. An example where you seen the endocytosis, Most common example like the amoeba. Now we are go to the question number 11. Where our genes are located? You know genes, where is present? What is the genes? Genes are the functional segment of the DNA. Functional means what type of function they do? Genes, they ultimately the basis of the heredity, and they ultimately involved in the protein synthesis in case any cell. You know, this is the nucleus. Inside the nucleus, the chromosome is there, and chromosome is the main site where the gene are located. So gene are located on the chromosome in the nucleus. Now we go to the next question. Chromosome number, and what is the chromosome number inside your cell? You have the many cells, right? But what is the number of chromosome present inside your cell? Please write down in the comment box. If you know, please you write down the answer. What is the number of chromosome present inside your nucleus in your normal body cell? Please write down. Next question. Name the two structures found in the plant cell but not in the animal cell. That means this is the two important structure found in the plant cell but it is not present in the animal cell. You know, the cell wall and the chloroplast. It is the most important two structure. It is present in the only the plant cell, not in the animal cell. Right? Now we go to the question number 13. Name the two structure found in the animal cell but not in the plant cell. Lysosome and the centrioles. This is a two important unique structure of the cell organelles which is present only in the animal cell but not in the plant cell. Right? Now I go to the next question, question number 14. Which organelle is involved in the formation of the lysosome? Which organelle are formation of the lysosome? That means these organelles are helped to the formation of the lysosome. The Golgi apparatus and got the Golgi bodies, right, which is involved in the formation of the lysosome. This is very important question. Now we go to the question number 15. What is the outermost layer found in the animal cell? In the animal cell, the outermost layer are the plasma membrane. Plasma membrane. Cell wall is not there. Cell wall is present in the plant cell. But animal cell, the outermost layer is the plasma membrane or the cell membrane, right? Next question. What is the outermost layer found in the plant cell? You know, the plant cell, their outermost layer is called the cell wall, right? Cell wall is the outermost layer. Next question, question number 17. Which organelle is the storage sac of the solid and liquid material? You know, the cells are required to store some materials, but in the cell, which structure is there where the all material are stored? Like the storehouse, this is the vacuole, right? Vacuole are the storehouse where the all the solids, liquids or the gas materials are stored on it. You know the what is the sometimes you know that the vacuole is a sac-like structure. It is covered by a membrane. But what is the name of the membrane which is enclosed the vacuole? Please write down in the comment box. Write down the comment box. So what is the name of the membrane of the vacuole? Now we go to the next question. Question number 18. Question number 18. Which organelles serve as the channel for transport of the material between the cytoplasm and the nucleus? Like, it is like a channel. It is helped to transport the material between the cytoplasm and nucleus. The structure is the endoplasmic reticulum, which is helped to serve transport the material between the cytoplasm and the nucleus. Next question. What is the microscope? What is the microscope? A microscope is an optical instrument. It is an optical instrument which is consisting of lens or the combination of lens. Sometimes you know the simple microscope is there, they have only single lens, but compound microscope they have more than one lens is there, and which ultimately renders the minute object distinctly visible. Sometimes the very small object is there which is you cannot see in the naked eye. That time we are use the microscope to see it, right? So the microscope is the optical instrument which is consisting of lens or the combination of lens, which help to see the minute object or microscopic object, which one we cannot see in the naked eye. Next question number 20. Why is light microscope is called the compound microscope? If you go to your laboratory in the school, that time you find some microscope. This microscope is a light microscope. Why it is called the light microscope? Because their illumination source is the light, right? But why it is called compound? Why it is called compound? Because this type of microscope consists two or more lens. So it is called the compound microscope. So in the simple microscope, they have only the single lens. Next question. Which organelles is associated with the ribosome formation? Which organelle help to the formation of the ribosome? This is the nucleolus. Right? Nucleolus is the structure organelle which is associated with the ribosome formation, not the nucleus. Nucleolus. Next question number 22. What are the chromosomes made up of? You know the chromosome where it is present inside the nucleus, but what is the chemical composition of the chromosome? Chromosomes are made up of DNA and protein. Histone protein also be there, but you write down as like the DNA and the protein. You know what is the full form of DNA? If you know, please you write down in the comment box what is the full form of the DNA. What is the full form of the DNA? Next question number 23. Define plasmolysis. Plasmolysis, it is like the shrinkage of the cytoplasm. Sometimes when a cell you are kept in the hypertonic solution, that time the water are exit out from the cell. That time the cell are sink. This process behind it, this is the plasmolysis. So plasmolysis is a shrinkage of cytoplasm due to loss of water when it is kept in the hypertonic medium. This hypertonic solution or hypertonic medium must be write down in your copy because it is the most important part. Next we are discuss next question. This is the what is the nucleoid? Nucleoid. So if you are consider any prokaryotic cell, they have no nucleus, so their genetic material where it is present? It is present in the cytoplasm. This nucleoid material is in the prokaryotic cell are called the nucleoid. It is a undefined nuclear region in the cytoplasm which is present in the prokaryotic cell. So you please mention the prokaryotic cell. It is must be written in your copy. Right. Next question. Which organelles other than nucleus contain the DNA? In the nucleus the DNA is there, but other some organelles is there in the cell structure which have the DNA. Right. The two most important organelles is the mitochondria and the plastid and the plastid which contain the DNA. Which organelles other than the nucleus contain the DNA? Mitochondria and chloroplast. That this is helping the photosynthesis. Next question number 26. Name the only cell organelle seen in the prokaryotic cell. Name the cell organelle seen in the prokaryotic cell. The ribosome, mainly 70S type of ribosome, this is the cell organelle seen in the prokaryotic cell, 70S type of ribosome. Only ribosome, if you write down, this is the wrong answer. 70S type of ribosome, it is present in the prokaryotic cell. And the ribosome is the cell organelles because other membrane bound cell organelle is not present in the prokaryotic cell, right? Eukaryotic cell, they have the lots of different type of cell organelles, but prokaryotic cell only the ribosome is there and they are the 70S type. Next question, which microorganism can be made into the crystal? So which microorganism can be crystal? If it is convert to crystal, which organism, which microorganism? This one is the virus. Next question, which organelle detoxify many poison and drugs in the cell? Sometimes the cell in the cell enter some harmful or poisonous substance. That time cell may be die. So one cell organelle is there. This is detoxify this poison or the drugs and protect the cell. This is the peroxisome. The very unique cell organelles. Next question, name a cell organelle which is non-membranous. It is must be most and most important question. The cell organelle which is non-membranous, that means it is not covered by the membrane, right? Ribosome is the example. The ribosome have the two subunit. One is the small subunit, another is the large subunit. But in the case of 70S, what is this two subunit is there? Please you write down in the comment section. Next question, question number 30, most and most important question also. That define the diffusion. The diffusion is the process. In that particular process, the movement of molecules are happening from region of higher concentration to region of lower concentration. It is must be mentioned in your copy. The molecules are moved from the high concentration to the region of the low concentration. It is called the diffusion, right? Next question, question number 31. Why is endocytosis found in animals only, not in plant cell, but why? That is most important. For endocytosis to occur, the outermost membrane should be flexible like the plasma membrane. But in the case of plant cell, the cell wall is there. This is the outermost membrane or outermost layer, but the cell wall are very rigid. They are not the flexible in nature. So endocytosis occur only in the animal and not in the plants. Now we are go to the question number 32. Which cell organelles control most of the activities of the cell? This is called the brain of the cell also. This is the nucleus. Nucleus, they are called the brain of the cell because they control the most of the activities of the cell. The nucleus contains the DNA. Already you know that what is the full form, deoxyribonucleic acid, which contain all of the information and they control all the activities of the cell. So the brain are called brain of the cell that is the nucleus. So this is the most important 1 marks question. So please you write down all this question and answer in your copy. This is the most and most important some questions. Now we go to the question number, next question, that is a 2 marks question, short answer type question from the fundamental unit of life from the cell chapter. First question, describe the microscopic structure of the cell. Describe the microscopic structure of the cell. So from that 1 marks question, now we go to the question number short answer type question. Please give me one moment, please. Short answer type question, 2 marks question. So every question carry the 2 marks. Now we go to the first question. Describe the microscopic structure of the cell. Describe the microscopic structure, that is you cannot see in the naked eye. So cork cell were the first cell to be observed. cork cell is the first cell which is first to be observed. And these cork cells are like the box-like compartment is there, forming a honeycomb-like structure. And cell organelles are found embedded in the cytoplasm. These are smaller in size and bounded by the plasma membrane. This is the overall microscopic structure of the cell. Next question. How can you calculate the magnification power of the microscope? I already tell you that microscope have the multiple lens, and magnification of the microscope is calculated by the multiplying the powers of the eyepiece and objective lens. Eyepiece is a one kind of lens and objective lens is another kind of lens. If you multiply these two lens power, that time you find out the magnification power. If the eyepiece is the 100x and the objective lens like the 10x, that time the magnification power is 1000x. Right? Very easy. Please bear in mind. Next question. What is the cell wall and how it is formed? Cell wall is a non-living. Please you bear in mind. It is a non-living structure and freely permeable. That means all molecules can easily enter or exit by the cell wall, and this is ultimately the bounding the plant cell. Cell wall is not there in the animal cell, and it is secreted by cell itself for the protection of the plasma membrane and the cytoplasm. Plasma membrane and cytoplasm, it is present inside the cell wall, so it is give some protection. Now we are go to question number 4. Why were the scientists not able to observe most of the cell organelles before 1940s? Because you know, before the 1940s, scientists could not view the cell only under the light microscope because they have some limitation. But when the electron microscope are invented, the time is the 1940. 1940 is a time where the invention of the electron microscope takes place, and which is enable the scientists to observe the cell in the greater details. Already you know that we know the total all the information about the cell. It is possible due to invention of the electron microscope. Other than we do not understand, we do not know the internal structure of the cell organelles. Next, the question number 5. There would be no plant life if the chloroplast did not exist. Justify. This type of question is very important in the CBSE syllabus. You know the chloroplast, which is contain a pigment. What is the name of the pigment? It is a chlorophyll. What is the function? It is responsible for the food preparation. What is the name of the process? You know, this is the photosynthesis process. And hence, if there were no chloroplast, then there would not have been not have been any plant life, because they are not able to photosynthesis and they are not able to produce their food. Right? Next question number 6. Why is the Golgi apparatus called the secretory organelles of the cells? From that question, we already understand that what is the most important function of the Golgi apparatus? That means have some secretion. But why they are called the secretory organelles? Because it is the packaging material is synthesized in the ear and dispatched to the intracellular plasma membrane and the extracellular cell surface target by the help of the Golgi apparatus. Next question number 7. What are the functional region of the cell? There are the 3 functional region of the cell. First of one, cell membrane or plasma membrane. Second part is the nucleus, and third and most common part, that is the cytoplasm. Next question. What is the cell sap and give its composition? Liquid content in the vacuole of the plant cell in the called the cell sap. Also it is defined as like cell have the water and some other materials which is dissolved in the cytoplasm. on the back wall also be there. This water plus other materials which is mixed together, this is called the cell sap. Cell sap contains sugar, amino acid, protein, minerals, metabolic of waste, or are mixed together in the cell sap. Next question, what is the cytosol and the cytoskeleton? Cytosol, you know, that is the semi-fluid part of the cell in the cytoplasm, which is embedded between the cell organelles. On the other hand, the cytoskeleton is a network of protein fibers which provide the supporting framework to the organelles. Clear? The cytosol and the cytoskeleton. Next question, what are the secretory proteins? Give example A secretory proteins. Proteins which is ultimately synthesized by the cell and released into the outer medium of the cell called the secretory protein. Like you know, the secretory protein include like mucus, digestive enzyme, like hormones, all are the secretory protein because it is produced inside the cell but it is released outside the cell for their function. Next question, question number 11. Why are peroxisome mostly found in the kidney and the liver cells? Peroxisome contain various oxidative enzyme which is detoxify the toxic material. Right? So blood carries the various toxic substance to kidney and the liver. A large number of peroxisome is present in that particular site for detoxify this type of toxic material. Next question, what do you mean by the plasma desmata? The cell wall, due to the presence of the cell wall, the exchange of material between the plant cell is not possible because this is very hard structure. Therefore, the protoplast of the plant cell are connected by a cytoplasmic channel. This is called the plasma desmata, by which the exchange of the material are happening between the two cells. So, plasma desmata mainly are present in the plant cell. Next question, why do the animal cell not have the cell wall? Animal cells do not have the rigid cell wall. Why? Because this is are incompatible in the animal cell, because the fluid cell membrane provide in the animal cell freedom to mobility and the formation of different tissue which is not present in the plant. You know that in the animal bodies the different kind of cell is there, they have the different function. It is possible for their flexibility. Next question, question number 14. Why are Golgi bodies are found in the large number of cells which is secrete in the digestive enzyme? The main function of the Golgi bodies, what is the main function of the Golgi bodies? To release the protein enzyme by the small vesicle. No other organelles have this property. So therefore, these are the largely present in the secretory cells because for their function. What are the significance of the pore present in the nuclear membrane? You know that in the nuclear membrane and some pores, but what is their function? This allows the transport of water soluble molecules across the nuclear envelope. RNA, ribosome, this is move out of the nucleus due to the presence of these pores, nuclear pores. Next question, what are the aster and the spindle fibers? Aster, you know the two star-like structure radiating fibers from the centrosome. It is happening in the animal cell. What is their function? They help to ultimately locating the spindle and trigger the cleavage of the cytoplasm. This is helping the cytoplasm. On the like a cell, this is a centriole is there, and they secrete some ray-like structure. This is called the aster ray. But when this type of fibers are produced, this is called the spindle fiber. Spindle fiber are formed from the microtubules during the cell division only. Next question, do you agree a cell is the building unit of the organism? If yes, explain why. any organism made up of various organ system, like the digestive system, like the nervous system, but these organ system ultimately are made up of various organ, which is made of tissue. A tissue are made up of cells, right? So cells, this is aggregate and produce tissue, and different tissues are aggregate to form organ, next organ system, next the organism. So it is the right. Next question, question number 18. If you are provided with some vegetables to cook, you generally add salt into the vegetables. After adding salt, vegetable release the water. But why? When you salt at that time, a hypotonic medium is created. Particular hypotonic condition means the concentration of the salt molecule is more outside, that means it is a solute, the vegetables, then in the inside. So due to the osmosis of water from the vegetables, the water is come out, right? Now we go to the question number 19. What are the chromatin, chromatid and the chromosome? Chromatin are the thin thread-like structure which is composed of DNA and protein. This is rod-like chromatid, like this is like the chromatin material, right? And when they are condensed and form a this like structure, it is called the chromatids, and this one is like chromosome. How the bacterial cell differ from the onion peel? Onion peel is a plant cell, or bacterial cell is the prokaryotic cell. So bacterial cell is very small, 1 to 10 mm, and the size is larger on the onion peel. Nucleus is absent, nucleus is present because it is a eukaryotic cell. It is prokaryotic and eukaryotic cell, and cell division takes place by the fission methods, and cell division takes place by the mitosis methods. Next we go to the question number 3 marks question. Now we go to the 3 marks question. The most important question, the 3 marks question, difference between the diffusion and the osmosis. This is the most important question. Diffusion occur in any medium, but osmosis occur in the liquid medium only. And in here, diffusing molecules may be solid, liquid or gaseous solutes. But osmosis, in that case, the involve a movement of solvent molecules only. Semi-permeable membrane is not required, but in the osmosis, semi-permeable membrane is required. Next question. Difference between rough and smooth endoplasmic reticulum. Rough endoplasmic reticulum, which is attached, ribosome are attached on their surface. Smooth endoplasmic reticulum means the ribosome are not attached to their surface. RER are help to manufacturing the proteins. On the other hand, the smooth endoplasmic reticulum are help to manufacturing the lipids. Next question. What is the membrane biogenesis, and how is plasma membrane formed during this process? The process of plasma membrane formation is called the membrane biogenesis. Following organelles are involved in the biogenesis process, like rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, secretory vesicles, and the plasma membrane are plasma membrane are produced. The protein and the lipids are first synthesized in the rough endoplasmic reticulum and the smooth endoplasmic reticulum respectively. These are then transported to the Golgi complex for their modification. After modification, this is transported to the cell surface through the vesicles which bud off from the Golgi complex, fused with the cell membrane, and a part of the new membrane are formed. This is called the membrane biogenesis. Write the name of the different plant part which are chromoplast, chloroplast and leucoplast are present. You know the chromoplast are present in the flower, fruit, the coloring part. Other than the green coloring part, chloroplast are present in the leaves and green part of the stem. Sometimes in the initial stage some stem color is green, that time it is due to chloroplast. Leucoplasts are present anywhere in the plant. They are colorless and the stored food, more commonly the roots in the site where the leucoplast is there. Next question, what is the cell division? Give the type of cell division. Cell division is a process by which formation of two or more daughter cells are produced. Cell division are most commonly the three types: mitosis, and meiosis. You know, the cell division also have the two phases. At the first phase, the nucleus division are happening. This is called the karyokinesis. After that, the cytokinesis are happening. Cytokinesis means the cell division. In that particular phase, the cytoplasm division takes place. So cytoplasm division means the cytokinesis, and karyokinesis means the nucleus division. Next question, what is the difference, functional difference between plasma membrane and the cell wall? Plasma membrane, it holds the cellular content and control the passage of material in and out to the cell, and it gives the protection, strength and rigidity. Same plasma membrane are semi-permeable in nature and cell wall are completely permeable in nature. Plasma membrane is not elastic, right, and cell wall comparatively elastic, and they are rigid. From that point of rigidity, the cell wall is more rigid, but from the elasticity in nature, they give some turgidity. What are the function of the nuclear membrane? You know, the nuclear membrane, which is separate the environment of the nucleus from the rest of the cell. What is their function? From nuclear membrane, their function is to protect the genetic material from damage, and it facilitate and regulate the exchange of material in and out of the nucleus. Next question, write a note on the Golgi apparatus and the function it is performed. Golgi apparatus, also it is called Golgi bodies. It is a membrane bounded fluid filled vesicle, vacuoles and cistern structure. In animal cells, you know, they are larger and only one or two in number. What is their function? They are involved in the transport and modification of protein, lipids and the carbohydrate. It is helped to formation of the cell plate during the cell division. It is involved in the formation of cell wall, and the material synthesized in the near the endoplasmic reticulum, it is packaged and dispatched by the Golgi apparatus. This is the most important question. How will absence of any one of the cell organelles affect the cell working? If you know, every cell organelles have a particular function. So function of all the cell organelles are interlinked to each other, ultimately working on the cell. So even a single link is missing, that time cell ultimately suffer and die. Next, draw a net diagram of the plant cell and label any three parts of the and differentiate to it. Right now, it is a most common picture. It is a plant cell, the vacuole is there, chloroplast is there and the cell wall is there. This is the most common structure, you know, that is present in the plant cell. This is the animal cell, very good picture it is, and so smooth endoplasmic reticulum, centriole, lysosome, right, nucleus and mitochondria. It is a animal cell picture. Please you draw this picture before your exam, because one of this picture must become into your final term exam, right? Next question. Now we go to the 5 marks question. 5 marks question, draw a label diagram of the mitochondria, write the function of the mitochondria. So please you draw this picture. All of this picture are most important. Plant cell, animal cell, bacterial cell, mitochondria picture. In that particular picture, mitochondria have the two membrane, outer membrane and the inner membrane. And outer membrane are in that particular, this side are called the matrix. In here some DNA molecules, and this is called the cristae. It is the structure of the mitochondria. Next question. Function of the mitochondria. It is the main site of the cellular respiration where the ATP are synthesized. Already we discussed another important question, what is the active transport and what difference between the active and passive transport. The process by which the molecules are moved fill against the concentration gradient with the help of energy or ATP, it is called the active transport. But what is the difference between the active transport and passive transport? It is involved in the movement of molecules against the concentration gradient, that means low to high. Passive movement involves the molecules along with the concentration gradient, that means high to low. Active transport require ATP and passive transport require no energy, no ATP. It is a rapid movement, and comparatively passive transport is a slow movement because it is not required the ATP. Movement of large molecules ultimately occur by active transport. Only small molecules or the water molecules only transported by the passive transport. Next question. A plant cell and under electron microscope and difference from the animal cell. So this is the most important picture of the plant cell. Please you draw this picture with correct labeling. In mitochondria, microtubules, vacuoles, right? Ribosome, nucleus, Golgi complex, cell wall, rough endoplasmic reticulum, vacuole. These are all the different parts of the plant cell. So plant cell have the chloroplast, plant cell have the large vacuoles, plant cell have the cell walls, which is not present in the animal cell. Right? So this is the most important some question about the fundamental unit of life or the cell chapter. So all these questions please you write down in your copy and bear in mind all these question is the most important for your final term exam. Thank you very much all of you.